Good afternoon. This is Derek Pizarro with AST Environmental. I have Ed Winter and Dwayne Guilfoyle with me here today. I appreciate everybody joining us. This is a trap and treat CAT 100 presentation, and we're giving a 5-year review. We'll get into those details in a little bit. So first, let's just talk about asking questions. We will be holding questions until the end, so please enter them in the chat or the question box. There are no polls for this presentation or handouts, so you don't need to concern yourselves with that portion of the dashboard. And feel free to type them in at any time, and we'll moderate them at the end of the presentation. So first off, before I hand this off to Ed Winter, Vice President at RPI Group, we just wanted to review places we've used CAT 100 in the last 5 years. And states and countries you see shaded in dark blue there are CAT 100 sites over the past 5 years that we've installed product and have data returned to us from the monitoring that we've conducted post-injection. But to give you a little history, this product, CAT 100, was developed in 2015 by Remediation Products, Inc. out of Golden, Colorado. It was installed into a site, the first one in Indiana, in 2016. And subsequently, you can see the footprint that we've created here with this product. It is used extensively in Indiana, New Jersey, North Carolina, and overseas as well. And the best way to think about CAT 100 is this product was initially designed to manage DNAPL percent level concentrations of contaminants, CVOC specifically, but there are some others we'll discuss that it can manage as well. But there are ways to design and install it for lower concentration applications, PRB applications. This isn't just something that can be used in a source zone. And because we have some flexibility on how we design and optimize this product for each site, this is also applicable for commingled plumes, not just a singular CVOC or another contaminant plume. So with that, here's just a brief intro of compounds that we have either completed remediation of in the field and are in a monitoring phase, or that we're currently designing bench test studies around. Most of these have actually been completed in the field at this point, and right now we're running multiple 1,4-dioxane benches to affirm how well that works. That's an emerging contaminant that we're heavily focused on in a number of states right now for specific clients. But the idea here is that we can, with commingled CVOCs and other contaminants of this range, we can do this in one step. This isn't a multiple injection, multiple product, combination of oxidants and reductants, et cetera, to get this done. This is a one product, one step application. So with that, Ed, Winter's going to take us into the first half. Ed, are you there?
I am. How are you doing?
Good. Thanks for joining us.
Yeah.
I'm going to hand you the controls now. So let's see how that works out here. All right. So we're just in the interim screen here, and Ed's going to pull up his presentation and present it. All right, we see your desktop.
So which presentation?
Third one over. Third one over is.
Yeah. Right, here we go. Thank you. And obviously the next question is, which one is presentation mode?
I think if you go to the upper right there, Ed, I see a button on my screen that says present. And we see it.
Okay, well, apologize, folks. That's my own failure there, but let's get started. So what is BOS 100? And BOS 100 is a part of CAT 100. So we'll have to kind of introduce that first to get the bigger picture. BOS 100 is a very unique product that is manufactured by taking activated carbon, running it to a very high temperature, about 680 degrees, with iron. And the iron becomes a chemical part of the carbon, such that it is not carbon and ZVI mixed. It is a unique product. The two, the iron and the carbon, are inseparable. In this particular graph, and this was really just, I'm presenting this to kind of show you a typical reaction where you see in the dark blue line, TCE, the TCE from the point of the injection of BOS 100 to the evolution of acetylene. And you'll see it happens very quickly. Now, what most people are used to thinking about is the evolution of ethylene. And that's because there's a kind of a confusion sometimes between the biological degradation of TCE and the physical degradation by non-biological means or abiotic destruction. So in the abiotic degradation, which is the pathway that'll be taken by BOS 100, you don't see the ethylene. And in fact, what you do is you see the double arrows pointing down towards the dichloroacetylene, et cetera, to acetylene. That is unique in the abiotic degradation pathway. That is the result of the iron within the BOS 100. Now, this is a very interesting experiment, and it's going to require a little bit of patience on the part of the audience. So let's see if I can pick up a pointer, and I believe we have a pointer here somewhere. Well, how about there we go, a spotlight. Ooh, see, isn't that nice? Okay, so here's where we start. We start with 75 milligrams of TCE and 150 ml serum vial. So if you converted that to a liter, it would be 400 milligrams per liter. You need to keep that in mind. So if we start off in one day of having 75 milligrams of TCE and 150 ml vial, and in one day we're going to generate 180 ppm of chloride, which is a 44% degradation. Remember, the 180 would be over 400 if it were per liter to get it in the ppm. Now, what happened is that we used to, when we would get the BOS 100 product in, each time we would get it in from the manufacturer, that it was a different manufacturing lot, we would retest it to make sure that it met, the QA, QC requirements. Not that we don't trust the manufacturer, it's just good policy, correct? So those serum vials, we'd push them aside in the corner of the lab and they begin to build up. And one day we said, oh, well, I wonder what's going on in those vials. Because we weren't really planning, initially we weren't planning an experiment. Now we've done this experiment multiple times since then, but pulled those serum vials. And that, in this particular case, is at 1,147 days later. And what do you see? The chloride generation. The chloride generation is 432 ppm, or essentially all the chloride, all the TCE has been turned to chloride. There's some error in any of these kind of measurements. That's why it's not straight 100%. So then we looked at that vial and we said, oh, well, after 1,000 days, not surprising, you don't have any more TCE. But what's the real question? The real question is the iron within the carbon, is it still active? So we said, well, let's find out. Put the serum vial, throw in another 500 parts per million TCE. Now understand there's only 5 grams of carbon in these vials. So, that's more TCE than we should ever be able to certainly sorb on carbon and certainly break down with the amount of iron that is in the BOS 100. So we did that a few days later, and at 10 days, what do we see? We see 31 parts per million evolution of chloride, which is about 8%. And at 60 days, right here, we see 135 ppms of chloride evolved, which is 33%. So what are you learning from this little experiment? What you're learning is that the capacity of the iron in the BOS 100 is greater than what you would presume it to be if it were just iron alone. Because right here at this barrier should have been the end of all the TCE that the iron would have been able to break down. Yet, when respiked, it continues to break down TCE. The iron is lasting longer than you would ever expect it to last, say, if it were ZVI. And that is the gist of that presentation and that experiment. Okay. So what do we want you to think about? You need to be wary of vendors that say they're going to mix activated carbon and ZVI together, and it'll be the same thing as a BOS 100 product. It will not be. The original patent that went to Robert Gillum for the mixture of ZVI and activated carbon was never commercialized, at least not by that lab or Robert Gillum. And the reason being is it doesn't work. Why? Because what you're hoping to do by mixing activated carbon and ZVI together, you're hoping to build a synergy, right? I mean, that's the only reason you would do it. You think, well, I got this sorption by the carbon and I've got this reaction by the ZVI. Oh, this ought to be great. And Robert Gillum thought so. But what you really find is you're not building that synergy. You're actually building a competition in which the activated carbon will sorb, say TCE, if we're sticking to that example, will sorb TCE 100 times faster than the TCE will even be able to react with the ZVI. So what you're actually doing is you're pulling the TCE into the carbon, you're keeping it from the ZVI. The ZVI itself has a limited lifespan. So you're working against yourself. And synergy is what you want, not competition. So the idea behind CAT 100, so now we're moving from the BOS 100, which has that iron as part of the carbon, as a chemical part of the carbon, to a new product called CAT 100. And CAT 100 wants to add to the BOS 100 the biological enhanced reductive dechlorination. And this kind of makes sense because microbes themselves love activated carbon. And there's a really good reason for that. Activated carbon itself provides a very unique surface in which you can lay down a extracellular matrix and in which a carbon biofilm can form. And thereby you end up building up a much higher microbial count on, say, activated carbon because of the porosity and absorption capacity and all of that, then you build up on the soil. And that doesn't really make too much difference what kind of soil it is. So BOS 100 has that unique property of the iron, and we're going to add to this a technology that we hope will be more than just the BOS 100 and the biological together. We hope we can do something that is superior, that is more than synergistic. Well, that is synergistic, right? Okay, so hopefully what we will be able to do is preserve the metallic iron. And you could think about, well, how would you do that? Well, if you actually produced more electrons than you were using to break down, say, TCE, you had excess, then you could keep the iron in a lower state, right? And that's what we were hoping to gain, something you could put in, you wouldn't have to come back, maintenance would be low, etc. So the key elements of CAT 100, it is activated carbon impregnated with metallic iron. That's the BOS 100 aspect. Then what we do is we add carbohydrates. Now, what are the carbohydrates accomplishing? As the microbes break down the carbohydrates, they release fatty acids, and then those fatty acids can be broken down and they release hydrogen. The hydrogen itself carries the electrons that can be used both to break down, say, your contaminant like TCE, and keep the keep the iron charged. So we have a set of microbes which are digesting the carbohydrate, driving that forward in a kind of overdrive reaction. And then we have a second set of microbes themselves, which are designed to degrade the contaminant and degrade the carbohydrate. I actually bobbled that a little bit. Two sets of microorganisms, one to degrade the COCs, the others to degrade the carbohydrate. And this is put together with the BOS 100 as a form of a single remedy. So this is biotic degradation, which most of you are quite familiar with. That, and I'll pick it up at TCE. You take TCE, you get DCE, vinyl chloride, you get ethane. And that's what you might expect if we had pure biological degradation. But let's see. So this is a CAT 100 bench test over time. Now, the bottom line, the gray line, is the control. The orange line is BOS 100 by itself. And you'd see that 68% of the iron that you would expect in the BOS 100 would be used around day 30, given the amount of chloride that's evolved. 80% of the iron would be used at 60 days, given the amount of chloride that would be evolved, and that's along the orange line. Also notice that the orange line, that once it jumps up initially, it more or less is a straight line. Now look at the CAT 100 line. In the CAT, that at 30 days, you would look, given the amount of chloride that's evolved, you would think that you had used 68% of the iron. If you look at 60 days, given the amount of chloride evolved, you would think that you'd used up 135% of the iron. Now, clearly, we can't use up more iron than we have. So you see there's something more going on. And part of that you can see. Notice when you look at the CAT 100 line that there's an inflection point a little bit before the 30 days. It's going up at one angle and it changes angles. If you have a single consistent mechanism, you usually do not change angles so abruptly. So what you're seeing is a different mechanism is starting to function. The first mechanism up to approximately or to the left of the 30-day arrow, say, it looks like 30 days, is consistent between the BOS 100 and the CAT, at least given the slope of the line. Post that, on the CAT 100, you see a second mechanism kicking in, and that's the biological. Now this graph is kind of a little bit the opposite of the other graph. So the green line, you see the CAT 100 again. You see the inflection point being pointed at on the left. But what if, and I'm going to go back, what if contrary to this graph where you had the BOS 100 versus the CAT, so the BOS 100 is the iron, it's the iron versus the CAT. We said, well, what if it were biological only? What would it look like? And that's the purple line. And you can see the purple line is less active than the CAT. So the CAT is a combination of the biological plus the reduction power of the iron combined, and is more than the sum of the two. Now, this is a little hard to follow, but not much. This is a carbon tet study. So if you look at the blue line, that is carbon tetrachloride and our initial concentration of 551 part per million. The next column to it, which is called 0.052186667, clearly an Excel fund there, is the carbon tetrachloride remaining. And this is, I believe, after 60 days. You also have, under the orange column, the initial chloroform generated when you break down the carbon tetrachloride. And the yellow column is the tetrachloride degradation, and then the chloroform remaining. So if you break down carbon tetrachloride, one of the things you get is chloroform, right? So you have an initial amount of chloroform in orange, and you have a later amount of chloroform in green, showing that you are breaking down the carbon tetrachloride. So you go from carbon tetrachloride in sulfate reducing conditions, for example, to chloroform. Now, where do you go from the chloroform? In, say, methogenic conditions, you will go to methylene chloride. And that's what you're seeing here in this graph. So the first blue bar is the initial methylene chloride. The orange bar is the methylene chloride that is being evolved as you break down the carbon tetrachloride. Now, the gray and the yellow happen to be tetrachlorofluoromethane, and that happened to be also present in these samples, and you see that it is broken down also as indicated by the yellow column. So what you're saying is we used TCE as our initial example to explain the BOS and the CAT technology, and then we go on to show you, well, here, if you look at carbon tetrachloride, we can also break it down. Now, CVI, for example, stops at methylene chloride, can't go any further. The CAT product will take you all the way to methane, CO2, and water. So the CAT 100 signature, in this particular case, it probably should have been higher in the presentation, but nevertheless, if you break down TCE, which is the red line, you'll see the TCE go down. You see the DCE climb initially, and then it starts to drop off. You see vinyl chloride climb a little later, and then it drops off, and that's exactly what you would expect, right? TCE, DCE, vinyl chloride. And then final, ethylene. So that would be, in this particular case, is the CAT 100 signature. Well, before we go on to this case study one, I had in a slide that somehow didn't make it into this presentation, but that's okay. And the slide had kind of a picture of a scientist on it, what he's all excited about. And we're doing a number of things in the lab. One of them has already been mentioned, where we have a series of bench tests on 1,4-dioxin, and they're going really quite well. So we're excited about that. We're not really quite ready to throw the data out into prime time, but it is going well. We've also looked at PCBs, And we're continuing to look at the carbohydrates that we use to charge the CAT 100. There are a number of things that can be done with the carbohydrates, and we're just really trying to optimize that because we believe we can do, perhaps we believe we can do much better. We'll see where the evidence takes us. And we're also branching out now and doing more biological work. And I hope that before long, we will be showing you some presentations that allow you to really dig in and see what's happening biologically. We have a lot of that in-house, but again, it's not really quite ready for prime time. But what I really want to emphasize, and one of the things I really liked about RPI and why I came to RPI, had to do with the fact that we spend a lot more time in the lab than we spend advertising, and consequently, we have some of these bench studies, and we're constantly trying to think of what can we do to improve and move forward. Okay.
Thanks, Ed.
Yep. Back to you, Derek.
As you teed it up here, we're going to look at some case studies, and then we'll take some questions. This first one, this was actually the first applied CAT 100 site in the US. It happened to be in Indiana. This was a site where we had significant DNAPL. Here's a little bit of the background. Manufacturing site, long time in operation, typically steel stamping, electroplating, machining, the typical part cleaning you would find at such a constantly operating historical manufacturing site. Lots of releases potentially to the ground surface. Also, there are underground process lines that may have leaked over time. So it was a fairly extensive investigation across the site here, and we'll give you a site map here in a second to show you a few areas we're focusing on. And those 4 primary areas, source areas, 2 degreasers, a scrap area, and a drum storage area. And our primary purpose here is to reduce CVOCs in soil and groundwater, both at the source location, but also to mitigate subsurface transmission and groundwater offsite. So we want to minimize offsite migration and protect potential offsite receptors. So here's a little bit about the conceptual site model. In the interest of time, multiple sources here noted. Here's the AST, here's our degreaser, here's our other degreaser. A lot of transport here going on the subsurface, moving from left to right. So we're dealing with a mix of ages of different weathered and breakdown compounds at this point. We have a lot to manage and investigate, so we had to complete an RDC. And if you've joined us for other presentations, an RDC is a remedial design characterization where we took, in this case, and take high resolution vertical soil samples. And by that, I mean, we typically take samples on a 1 or 2 foot basis, moving through the borehole. And that helps us design a remedy that both accounts for the mass that's in soil, which we have to manage, and also to what's desorbed and present in groundwater as well. So to handle that soil and groundwater fraction, we started off with the RDC in 2014. This was comprised of 11 borings and 1 groundwater grab sample. That promulgated a pilot, which took place the next year. And we're going to focus on this IRM area here in orange. Let me turn my pointer on too. There we go. So this is the area we're focusing on. We've got transport from top to bottom. This is offsite housing. Here's a street, and here's our boundary line right here. So we've got both monitoring points on site and offsite, showing we've got both source and transport here. And we'll get into some of the data here. We're going to look at 2 of these wells specifically to show you the enhancements that CAT 100 applied to this area allowed us to see significant accelerated CVOC degradation, both from a source and also in a PRB setting at that boundary line. The other thing we did during the pilot is we took that opportunity to enhance the original RDC completed a year earlier, and this was comprised of some additional borings and groundwater samples and samples from the existing monitoring well network at this area. So our goal, again, we want to achieve contaminant migration control from the area we just showed you. We're talking significant CVOC concentrations in both soil and groundwater. And the reason we chose CAT 100 is because of those concentrations. As Ed mentioned, A lot of the times we are examining CAT 100 for a site, it's because of source area CVOC concentrations, significant concentrations where you would typically be limited in iron applications, as in iron you could physically install in the ground as a starting point, or you're going to be limited in terminal electron acceptors or other bio necessities that would require expeditious breakdown to manage all the CVOCs we have here at this location. So let's zoom in on the area. The subsequent slides are going to focus on monitoring well 71 and 63. So we have our up gradient and source area, and here's our boundary line and eventually a PRB install. This area is about 5,400 square feet. We put in 215 direct push points. That sounds like a lot, but because we inject through pre-strung geoprobe rods, we're not limited by how much we can pump because our pumps are running at very high GPM, high pressures. So our pump run times are on the order of 10, 15, 20, 30 seconds. So we're really only limited by how fast we can probe. So while 215 points as a pilot, or in this case an IRM measure, sounds like a lot of area to cover, it actually goes pretty quickly. And most of this was completed after a January start. We wrapped up in early February 2016. So this did not take too long at all, despite having a lot of points. So what does the data look like after 6 years? Well, and I think, I can't remember, Ed, if you showed MW71 or 63, but the trend is relatively the same at these locations. We see a significant, in this case, parent TCE concentration, which was over 80 ppm, dropping off precipitously. That's again the carbon managing the mass we're dealing with. And then again, as he showed you in his slide here, you see the sequential breakdown as we both go through the sequential biotic process and also the removal of mass from the soil. Again, that spike in ethylene generation and a trend downward in CVOCs shows that, a starting point here of 80 ppm and trending downward to here of less than 1,000, a lot of concentrations, is a significant step moving from 2015 to current time frame. Again, the mirror image of chloride generation, one of the markers we use that Ed talked about in addition to acetylene for such a setting. And then here's the biological signature for MW71. Kind of a threshold here at 1 times 10 to the 4th here. That's our cell count. That's really where you can start to say in this setting that you're getting really good microbial activity and growth and managing a lot of the CVOCs you're dealing with in the subsurface. Some of the things to point out here, we don't have DHC in our blend, but we stimulate significantly even natural microbial populations through this process. Again, this is not just a one-step product that goes in, but a lot of the enhancements that are included in CAT 100 enhance what's already going on naturally in the subsurface. And here's the other well there. This is down gradient in the PRB setting at the site. So this is managing our off-site flux. And what we see here is a similar trend in both input TCE and DC at this point. Again, we do have more breakdown products in this vicinity due to the plume. But again, you see that downward trend and then eventually the ethylene generation. And as we start to run out of CVOCs, that decreases as well concomitantly. Biological signature for 63. Again, similar to what we saw in MW71. Again, we're getting good growth. We're producing a lot of bacteriological activity here, and we're even enhancing a lot of the other constituents in the subsurface to promote degradation. So the next one I want to go to, this happens to be in Europe. Excuse me, I tapped that too quickly. This is care of our European partner, Elskov. This is a presentation that Giorgio Siriani gave a couple months ago, specifically just to this site. If you're interested in a deeper dive, this one's also available on both AST and RPI's websites. But what we're going to look at here is significant concentrations of CVOCs, and we're going to use CAT 100 to manage most of this. And what we find here is in our upper right here, this is our soil PCE concentration. This again came from an RDC process that was conducted by Elskov. And then we're marrying that up here with the groundwater network. These are existing wells and some hydro punch. And this again is showing you the difference between soil and groundwater concentrations here because of an RDC process. So develop a injection program to develop the loadings we need for that. We need to couple these two up. I think on this next slide, we'll show you why that's so important. Okay, sorry, I thought I had a slide here. Initially, something we considered for this site was actually to do an L-shaped PRB here. I'm gonna go back to this slide here. Considered an L-shaped PRB here because of the groundwater concentrations. But when we saw the soil concentrations where most of the source resides up here, and again, this is a former dry cleaner that operated since the 60s, we still found most of the source material up here near the building. So that PRB that initially seemed like a decent idea here based upon just groundwater, again, the problems with just looking at groundwater and not doing an RDC, that altered the approach for this site. So the PRB was not applied in this L-shaped corner like we initially discussed. So here's our PCE plume. Talking about a 5-year difference here. Here's what we started with. Again, we're focusing here on groundwater as our monitoring and compliance point. And all this is conducted by taking VOA vials and sending them to RPI or a registered lab. That's what you can do with your sites. Here we use utilizing RPI's in-house testing and optimization lab. That's the lab we use for most of our RDCs. And what we're seeing here is the difference pre and post injection of PCE. And then here we're going to look at ethylene generation. You can see we start with very minimal activity at the site going on naturally, and the enhancements to that degradation and subsequent ethylene generation post installation of CAT 100 as we convert PCE. So how else can we look at actual removal, not just the dip in concentrations of CVOCs and the generation of dissolved headspace gases? Well, we can also look at trends in DNAPL. This is a graph that Elskov has prepared for us. We're looking at decreases in NAPL and volume removal, which they've calculated here as well. But we also can focus on biological activity. By providing those samples to RPI's lab, we can focus on identifying pathways that we're breaking down these compounds, not just seeing them disappear in the groundwater samples per se, but also be able to account for the why and how that they are disappearing in the subsurface, showing subsequent treatment. Here's some more of that data. This is our source zone. So X002, that's our source zone treatment. Again, very similar trends to what you saw on the previous slide, post-injection. And here we are mid-plume. This is MW63, not too far from the building footprint. A little bit less to start with, but again, very similar trends. And here's some animations that they prepared through the RDC process here. Get off my pointer here. So through that RDC process here, we're going to talk about PCE plume development, and this is what we're looking at post-injection of the CAT 100. So by development, we actually mean the reductions in the footprint, the concentrations of PCE in this case decreasing over time. So there's definitely a regression in PCE in that. And here's another view. This is also available in 3D in one of their presentations as well. Oops, come on. All right, and so here again in reverse, here's our ethylene development through this breakdown process. Just looking at the other side of this equation. Again, all this was generated from the RDC process, which was about 255 soil samples. We did discover a new source zone area that expanded the source zone area while we were up towards the building footprint. And most of this NAPL that we're dealing with in the animations resided about 3 to 6 meters below ground surface. All right, so as one last note here for this site, again, we did focus on a microbiological study for this client. There are more details to this side of the equation on the video that Georgia presented on our website. But I want to focus on one last case study here before I wrap up. This is a confidential industrial property site that we worked on with CAT 100. While this site is still active, we do have some challenging geology. So we completed an RDC in 2013. We focused on that. This one, 186 samples in total. Some more groundwater samples. It's always a good deal to couple up groundwater and soil samples if you can. That shows the partitioning between both soil and groundwater, which is critical to understanding where to place reagent and at what loadings. For this site here, we selected a PRB, but also this is a combined remedy as well, because we coupled up BOSS 100, CAT 100, and sodium permanganate through a soil mixing program. So from our RDC, we identified a DNAPL source area. We have both unsaturated and saturated impacts. That's why we're doing a combined remedy here. Again, the BOSS 100 and CAT 100 you saw on that previous notations, the previous notations on that last slide. The BOSS 100 and CAT 100 products we've dealt with here do need groundwater present. So they use groundwater as a transport mechanism, so they won't function well in the vadose zone. You need another way to attack those. And so that's why we use the combined remedy at this site. So here's the source zone application we want to focus on. This was completed in April 2020, 7,000 pounds of CAT 100. And you can see there on the map on the right, we have the CAT 100 injection area as a standalone treatment cell. And then we have our overlapping BOSS 100 and groundwater and soil mixing in the vadose zone upper towards the source area footprint there in the upper right. And so here's our results from monitoring that data. Very similar signature. We're going to focus just on the CAT 100 footprint towards the boundary line of the site. Again, TCE, breaking down DC, VC, and eventually we could see that we're getting these concentrations to drop, and that coincides with a spike in ethylene. And then here's a molar plot of concentrations post-injection. As you can see, we do get that sequential breakdown again, eventually trending down to very low concentrations. And one last look at MW18. Here we're focusing on carbon dioxide and chloride generation to prove out that treatment. So we're at 12:45, which we budgeted for the entirety of this presentation. So we're running a little bit long. I do apologize for that. We do have a number of attendees here. There are some questions in the chat, and I'll bring Ed and Dwayne in for these as well. If you can stay, much appreciated. Feel free to enter questions in the chat box or the question box, but I'll open the floor up to them and feed them the questions. You guys off mute?
Yes.
Excellent. So Ed, first question here for you. Could you discuss the difference in the signatures seen in byproducts generated by traditional BOSS 100 versus CAT 100, specifically an abiotic versus biotic signature between the two products?
Okay. So the BOSS 100 would be an abiotic process in which you are using the zero valent iron is donating the electrons to basically tear the chlorides off of, if we stick with, say, TCE, and you'll end up with generally acetylene. That would be a pure abiotic reaction. In the CAT, let's set the CAT 100 aside and just say, what if you had a biological reaction? The biological reaction would be the one that each of you are fairly familiar with. And that's where you go through DCE, vinyl chloride, and ethylene. So there's a clear difference in mechanisms in the way they look. Now, what CAT is doing is combining the two. You have the iron present within the CAT, but you also have the carbohydrate. And a lot of you are probably familiar with people injecting like edible oils, right? And what happens, the edible oil breaks down and you end up producing hydrogen. Well, the carbohydrate's doing the same thing. The microbes are breaking down the carbohydrates to fatty acids. The fatty acids are broken down and you get hydrogen ion. The hydrogen ion is then the electron donor, in that case for tearing the chlorides off TCE, but it is also eventually you produce so much of that that you are able to keep the iron in good condition. So that's the comparison between the two. Now, if there's a more specific question, I'll be glad to take that.
Sure. Well, we can even dive a little further on this. There are two other questions here that may relate to that. The first being, would CAT 100 work in both aerobic and anaerobic environments?
Yes, and the reason being is what you hope to do, and it's not just our products, it's anybody's product pretty much, that you want to dominate the environment in the aquifer in which you are treating. And so when you put in a lot of carbohydrate, you're going to basically you're going to drive that aquifer towards being anaerobic and reductive. And that's what you want to do. So it doesn't really make too much difference where you start as far as the aquifer being aerobic or versus anaerobic, I should say.
And kind of continuing with that, could you speak a little bit about preventing secondary plumes of DC and VC as these products break down?
Well, part of the value of the carbon itself is the absorptive capacity. So you're able to hold back plume development. And that's really the beauty of carbon itself. Now, if that's all you had, if you only had carbon, you could slow the development of any plume by the sorptive capacity of the carbon itself. So that prevents a lot of secondary development. Now, when you go beyond the carbon to actually having a mechanism that breaks down the contaminant, it's important that the mechanism that you have is robust enough to drive you all the way to the end product, whether that product be ethylene or just methane and CO2, depends on what contaminant we're actually starting with. So that is important. And in the case of CAT, it certainly is robust enough, as you saw in multiple graphs shown to you by Derek, that we drive this reaction to the end. Now, if you only had, and I hope this is not confusing that I bring it up this way, but if you only had ZVI and you had carbon CAT, basically you'd end up with methylene chloride and wouldn't be able to get any further. And you saw, at least in the one reaction, I was able to show you that we're able to drive that all the way to methane, CO2, and water.
Great.
I'd comment on that too, Derek, that if in the case of a high flux, site that, we've often looked at combined remedies where, it's source treatment with the CAT 100, but also providing prevention for migration if the flux is large enough to have VOC 100 by itself, on the perimeter or in a PRV scenario to be able to manage if there are any daughter generation that isn't able to be controlled by the carbon itself within the CAT 100, that you've got a preventative method to be able to capture that and control that from migrating and developing a plume further down the gradient.
Yeah, that's really important, Dwayne, because what I tried to answer the question from was basically just from more or less the biological point of view. But for any of you out there who have a site, when you design a remedy, there's that engineering and geological aspect and not just the biological aspect, and you have to put that together as a whole picture if you want to control plume development.
Okay, and so, yeah, go ahead.
No, that's it. Go ahead, Derek.
So we've actually got two questions here. I'm going to try and distill them down. Actually, there's three questions, kind of in a similar vein. Is there a maximum concentration or a minimum concentration of contaminants that CAT 100 could manage, specifically in PRB settings?
I'll answer the minimal. So we have applied CAT 100 in low dissolved plume scenarios in a PRB setting. And when I say low plume, I mean less than 100, 100 ppb and lower, where you're trying to get to, say 1 ppb cleanup standard. And really, again, like Ed said, the power of the carbon plays a critical role because it has the ability to accumulate mass. And by the ability to accumulate mass, it can handle biological degradation better by providing a larger food source versus if you were just trying to stimulate and clean up a low concentration site with just ERD alone. And Ed can probably speak better to that for the biological side, but the carbon plays such a critical role to be able to help do that. And often we look at the CAT 100 sometimes in PRB scenarios where we want to provide that longevity beyond what the iron can provide itself. So it's a combination, like Ed said, of looking at what are you trying to get to and how quick do you want to get there. But the carbon does play a critical role in not only a low concentration setting, but also in a high concentration setting to be able to manage the amount of mass that the bacteria actually are seeing, because the isotherms of the carbon will only allow the carbon to absorb so much mass. And what we're talking about here as far as the mechanisms for treatment are occurring on the carbon and not in the formation itself. Ed, do you want to add anything to that?
It's actually difficult, but I just won't resist a chance to talk. There's probably no maximum, except for just practicality in the engineering and the design. Obviously, if you have enough concentrated contaminant in one place, that is toxic to the microbes. You can reduce that toxicity with carbon. At the same time, in your engineered design, there may be times when you want to remove some of that free product before you move forward. And that's all geological. That's geology you're working in, that's engineering, and just looking at your greater picture of what you're trying to accomplish. So my first answer to repeat it is there's actually no limit except for the design itself and the goals that you lay out for yourself. So that would be my response, I think. Derek?
Yep, and maybe you could talk a little bit about the mixture again. There is a question about sustaining, and this would apply to either max or min concentrations, but how do we sustain this continuous process?
Okay. So where shall I start with this? If you look at carbon, if you could shrink yourself down to the size of a microbe and you were to walk up to a carbon molecule, it would look a little bit like, well, maybe an apartment building with the face ripped off. You have all these layers and shelves and places to live and places to be, such that soil itself doesn't compare. If all things are equal, the amount of nutrients, etc., are all equal, there's far more space in the carbon, and the carbon is far more advantageous to microbial life than the soil itself. That being so, if you get a good start on your microbial population, the microbial population will do well on the carbon. And as it grows on the carbon, it builds a biofilm. And in that biofilm, it stocks away early a lot of things it needs. So when other microbes die, they don't like throw them away. They're actually hung up there in the biofilm. Just think of it like an automobile scrapyard. So when you first come in with Cat 100, you're throwing in a lot of starch, and that starch is being broken down. And you're getting these fatty acids. The microbes are just booming. They start building for themselves kind of microbe cities, these biofilms. And by building the biofilms, they actually stabilize their environment. So now, they can survive toxic blows as a group far better than they can as individuals. They can survive a certain degree of desiccation far better than they can as individuals. And they can share products of metabolism with one another so that as a group, they can metabolize things that they can't metabolize as individuals. So for instance, there are a number, Campbell is one of the papers that come, there are a number of journal articles where TCE is broken down by microbial communities that contain no dihalocoides. And they do that because the community itself is greater than the individuals. So this is what you're trying to build with the Cat 100 onto the carbon is these communities. And so far as the best we can tell, I think the one site we have is like 5 to 7 years old. The communities still seem to be sound and still seem to be functioning. So if you can, and you think about it, in the subsurface, you are primarily carbon limited, not so much nitrogen and phosphate limited. Microbes aren't plants. So if they can sock away enough hydrogen, enough nitrogen in the form of amino acids and other things like that early, then they can continue to recycle those going forward. So you don't have to keep, feeding them and tending them and taking care of them. They will be able to take care of themselves because they have the carbon source, and it's the carbon source that's limited in the subsurface. So that was probably a much more complex answer than you guys were asking for. And hopefully I didn't ramble and it was clear.
No, that was great, Ed. I actually enjoyed the apartment analogy. So
everybody's got to live somewhere, right?
That's right. So we're at the top of the hour. It's 1 p.m. Eastern. I just wanted to thank everybody that joined us here and spending some time with us over the lunch hour on the Eastern end, or if you were elsewhere in the world during your busy day. So thank you so much for your time. All three of us, our contact information is up on the screen, or you can visit us at either of our websites and we can assist you with questions or a site you may want us to look at. So thank you again and hope you join us for our next webinar.